Hello. So today we're going to be working on the Mitosis Lab. So first I want you to think about experiment design. If you want to design an experiment to test whether a particular chemical increases the rate of growth of a plant, hopefully it's pretty obvious that you would put some in water and some in water with the chemical. So some with the chemical, some without. But the question really that I want you to think about is how would you measure the rate of growth? We want to be a little more scientific and sophisticated and precise than just measuring the height of a plant, like maybe you would have done a couple of years ago. So think about that a little bit. Hopefully you realized that a plant grows because it's going through mitosis. And so if we look at the places in a plant that are actively growing, like the very, very tip of a stem or the very bottom of a root, you should see a lot of mitosis happening. And the faster it's growing, the more mitosis there should be. The setup for this lab, first of all, this is not an experiment we're actually going to do. We don't have the time to actually grow plants and then make our own slides. And it's actually very tricky to make good slides. But this is kind of the context and the procedure that you should be thinking about as we look at data in this lab. If we took a plant like an onion, that's the one we're talking about in this lab, and we grew some of it just in plain water and some in water with a chemical mixed in, like you see in that picture there. You can then take samples of the tips of the roots where it's growing. You just cut off the root tip, you treat it a little bit to preserve the cells, dye it with a stain so that the chromosomes and the cell walls are visible, and then you put it under a cover slip and kind of squash it a little to spread the cells out. You end up with something that looks kind of like this middle picture, which might look familiar. We looked at slides very similar to this when we studied mitosis in 10th grade. If you want to know more about that procedure, you can click on the link at the bottom of this slide in the actual slides posted in Classroom to see how those slides are prepared. But the important thing is that once we have a slide of it, you can look at each of the cells in there and determine how many of them are actively dividing, are in one of the phases of mitosis, and compare that to how many of the cells are in interphase. And if you get a higher percentage that are dividing, then that root tip is growing more quickly. And if you have less that are actively dividing, then it's not growing as fast. The first question in the Mitosis Lab asks you to design an experiment. And when you do that, I would like your design to have a protocol similar to what we just talked about. It should be based on looking at slides and counting how many cells are in mitosis. The reason that's important is later on in the lab, it will give you some hypothetical data that you might have gathered from that lab, and it makes a lot more sense if your data matches your procedure. So write your experiment, design it, think about all of your constants and your variables and all of that, but have it be based on counting the number of cells that are in mitosis. The lab that we're doing looks like this. This is that first design and experiment that I just told you about. You can read that first paragraph to read a little more about the specific chemicals that we're talking about here. The part that we're focusing on in class today is the data collection. So this has to do with identifying the phases of mitosis and looking at some actual slides. So if we go to our next slide here, we're going to practice that just a little. Take a look at these pictures. They are mostly in order, so that should help, but identify for each one what phase of the cell cycle you think that cell is in. Hopefully you said the first couple are in interphase. In these two, you can't really make out specific chromosomes. The nucleus is all pretty solid colored. There's beginning to be a tiny bit of space between them in number 2. So if you said that was really early prophase, that's okay. By 3, 4, and 5, I would definitely call that prophase because you can see the chromosomes are condensing in these nice dark lines and there's lots of space between them, but they're not really organized yet. 6 is definitely metaphase. You have that nice solid line down the middle. Remember, the part that lines up are the centromeres of the chromosomes that have the kinetochores with the microtubules attached to them. That's the part that actually lines up the middle. The arms of the chromosomes just kind of flop all over the place. 7 is maybe late metaphase as the sister chromatids are just starting to separate. And by 8, we're definitely into anaphase as those chromosomes pull apart. 9 and 10 are showing telophase and cytokinesis. In pictures, there's no visual difference between telophase and cytokinesis because they happen at the same time. Telophase is technically what's happening with the chromosomes in the nucleus. So the fact that the chromosomes are kind of gathering at the two ends and a new membrane is forming around them, and you can see that the chromosomes are starting to unwind and uncondense here. Cytokinesis has to do with the division of the cytoplasm. So even in number 9, you might be able to see the very faint beginnings of a cell plate, and you can definitely see that in number 10. So as you collect data, you will be looking at some pictures on this website if you are at home. This has 4 different pictures that are very similar to what you would see if you looked through a microscope in class. Use those to practice identifying these phases, and also use one of those to make some sketches. You need to pick out 4 cells that are in 4 different phases, and for each one, label any structures you can see, cell wall or chromosomes or the nucleus or whatever is visible at that phase, and identify the name of the phase. And you have to insert a photograph of your sketches right there. The other thing you need to do using those pictures, pick out one of those 4 pictures, and count how many cells are in each of the different phases. I know there's a lot of cells on there. Be as accurate as you can. Probably you should actually count how many are in prophase, metaphase, anaphase, and telophase or cytokinesis. But you can estimate how many are in interphase. Just try and make it as accurate an estimate as you can. The data that you collect here should also be added to the spreadsheet that's linked in Google Classroom as part of the class data. That you will need to use. I will be totaling up all of the class data over the next few days as people enter it, and you'll be using the class data to perform an analysis for number 5 to figure out whether these prepared slides and images that we're looking at represent kind of the standard amounts that we would expect for the number of cells in interphase versus the number of cells in mitosis. The rest of this lab can be done, all except question 5, the rest can be done without the class data. And so you can continue working on this lab over the weekend. This is a pretty long lab, which is why I gave you a week to do it. But please don't wait until Thursday night or you will regret it because it is pretty long. And I want to make sure you have time to ask questions about chi-square or about making graphs with error bars. Those are things we haven't done for a while. So as you work through this lab, please make sure you contact me if you have any questions. I hope you have a great day. And let me know if you need help with anything.